Well, good evening. I'm Rick Lifton, the president of Rockefeller University, and I'm absolutely thrilled to welcome all of you to one of my favorite events of the year, the presentation of the Meister Greengard Prize, an international award that honors exceptional women in biomedical sciences. This is the 15th year the prize has been awarded to a total to date of 19 remarkable women who have made major contributions to biomedical research in diverse fields, including molecular biology, cancer research, immunology, biochemistry, genetics, biophysics, and neuroscience. Discoveries made by our prize recipients are among the most remarkable advances in the past half-century by either men or women. And tonight we're here to honor another amazing scientist, Dr. Jennifer Doudna, for her incredible discoveries about RNA structure and function. Dr. Doudna was chosen by members of the Prize Selection Committee, a group of esteemed scientists that includes 4 Nobel laureates. You can find the list of our selection committee in the back of your program. When Paul Greengard received the Nobel Prize in Medicine in the year 2000 for his crucial discoveries about the mechanisms of signal transduction in the brain, he and his wife, Ursula von Rydingsvard, donated his monetary share of the Nobel Award to establish tonight's prize, which is named in memory of Paul's mother, who died giving birth to him. Paul and Ursula's vision and generosity inspired Friends of the University to provide additional funding. In addition to the prestige of this award, this prize includes a $100,000 honorarium to the recipient. Paul and Ursula created this award based on their observation that women in science were not being recognized at a level commensurate with their scientific contributions. They were determined to use Paul's prominence as a Nobel laureate to address the issue by drawing attention to women scientists doing groundbreaking research. Now I'd like to share with you a short film about the prize that was produced by Paul and Ursula's daughter, Ursi von Rydingsvard-Grieve. Please enjoy the film. It is amazing that we've been giving this prize for 15 years, and I think it's a consequence of a visionary idea by Paul and Ursula originally, and the very high standards with which the recipients have been selected.
And now Professor Paul Greengard.
The genesis of this prize goes back to the year 2000 when I won the Nobel Prize in Physiology of Medicine.
It wasn't until he got his Nobel Prize that we thought this is a bucket of money that we can put to something that's really important to us.
The prize is called the Meister Greengard Prize because it was named in honor of my mother, who died giving birth to me, and it seemed that it would be a nice thing, since I wanted to do something about discrimination against women, to name it in her memory.
The Meister Greengard Prize is a real gem here at Rockefeller. One hope from this prize is that women who are fascinated by science, who are thinking about careers in science, will see these incredible women who have been leaders in science, have been successful in science, and are recognized in science, and see these women and say, I want to be like her and I bet I can.
I've always felt this affinity for this award because of the spirit of what a mother does for one. My mom died when I was in college, when I was 21, and so she never knew that I became a scientist. She never knew how happy a life of discovery is.
It's a great honor to get a prize, but to me the impact of the prize is far beyond that. To me the impact is a young scientist in a lab today. See that the honorees started just like them in a small lab, knowing very little, having no idea what they're doing, but they had a passion and something they're going after, that they will be inspired to do the same.
When I received the news that I was receiving the Greengard Award, I was deeply honored. Paul Greengard is one of the real pillars of science. Also, I thought it was fascinating that he wanted to honor his mother by highlighting the work of scientists, and women scientists in particular, as this prize is doing. Will encourage more young scientists, or more aspiring scientists, or even students who don't quite know what they want to do yet, to get involved in science. We need clever people from every walk of life, from every type of background, to get involved if they're interested, because I think when you have that kind of diversity, then you have diversity of ideas as well.
That was just great. Now I would like to introduce our presenter this evening. Each year, the Meister Greengard Prize is presented by a woman who, like the prize recipient, has made important contributions to the betterment of our society and the world. Tonight's presenter, Agnes Gund, is often referred to as the grande dame of the art world. She has not only helped to make the world a more beautiful place, but has used her considerable influence to make the world more equitable as well. Agnes Gund hails from Cleveland, Ohio. The daughter of a successful banker, Agnes felt that her affluence carried with it great responsibility to help others. A collector of modern and contemporary art, Agnes has held many leadership positions. She served as president of the Museum of Modern Art from 1991 to 2002, and currently serves as president of MoMA PS1. She has also funded programs at MoMA that create opportunities for members of underserved communities to have access to MoMA's spectacular art collections. Agnes is also a staunch advocate for education, women's issues, and environmental concerns. She was also a member of Rockefeller's inaugural Women in Science Committee and has been a steadfast supporter of the program here at the university ever since. She's served on the boards of other important organizations such as the Aaron Diamond AIDS Research Center, the Andy Warhol Foundation, the Barnes Foundation, Chess in the Schools, and the Frick Collection, the Fund for Public Schools, and the Robert Rauschenberg Foundation. She also serves on the board of the Cleveland Museum of Art in her hometown. One of her most extraordinary and enduring legacies to New York City is the remarkable Studios in the Schools program, which she founded in 1977 at a time when the arts had been virtually eliminated from the city's public school curricula. The program brings artists into New York City schools to teach children the importance of creativity in their lives. In the past 4 decades, a remarkable more than 1 million students have benefited from this program. And just last year, Agnes stunned the art world when she sold a Roy Lichtenstein painting from her personal collection. She used $100 million of the proceeds to create the Art for Justice Fund to support criminal justice reforms that reduce mass incarceration and rehabilitate men and women in the prison system. Founded in partnership with the Ford Foundation and the Rockefeller Philanthropy Advisors, Art for Justice has already committed more than $32 million in grant funding with the goal of disrupting cycles of incarceration. The fund supports a broad range of organizations, including grantees working to reform the bail system, end hiring discrimination against former prisoners, and sponsor art programs designed to keep young people out of jail. Agnes's extraordinary career has been well recognized by a string of exceptional honors. She was awarded the National Medal of Art by President Bill Clinton in 1997, and in 2016 she was elected an honorary fellow of the Royal Academy of Arts in London, which was founded by King George III in 1768 before he went on to his better-known role starring on stage in Hamilton. Sorry. Agnes has received honorary degrees from Bowdoin College, the City University of New York Graduate Center, and Brown University. And I'm delighted to add that just last week, Agnes was also awarded the prestigious J. Paul Getty Medal for her extraordinary contributions and support of the arts. It is our great pleasure and honor to have Agnes Gund with us this evening. Please join me in welcoming her to the podium.
Thank you very much. Thank you. Thank you again, Dr. Lifton, for that generous introduction. I extend my thanks to you and the members of the selection committee for inviting me here today. I would also like to acknowledge the presence of a very good friend and Rockefeller trustee, Marnie Pillsbury. As you know, she worked with David Rockefeller for many years. I am thrilled and proud to be here to help present Dr. Jennifer Doudna, with the 2018 Pro Meister Greengard Award. It is a very special honor for me to be here at this storied place where biomedical research and cutting-edge science happens. It is also a special opportunity for me to be here. Many of you in this room, your lives in science, facing its questions and its aspirations and its mysteries and its depths on a daily basis. But for many others of the science is not really understandable, not familiar, not known. For me, being here tonight provides an opportunity to acknowledge the efforts this institution makes to address the distance between scientists and us others. I know, Dr. Lifton, that the Rockefeller University has played a very special role in public life, not only because it advances science, but because it opens science to a broader understanding. The consciousness of this place, its conscience, if you will, has really impressed me. Your programs for parents, the institutes for high schoolers, the fellowships for college students, the art offerings, and here lectures are so much else. You do provide an open door at this university. Your Women in Science program, which inspires so much of us, is unique. And this prize, this special acknowledgement of women in science, is more of the unique ways in which a university connects science to society. There's so much happening here to show how seriously the Rockefeller University believes that science must be perceived as a public good by the public. I will talk briefly this evening about myself and my concerns in life because I have been asked to do that, as have earlier speakers at this prize celebration. I will talk about Dr. Doudna, whose evening this is, but first I want to talk about the prize itself, which was created by my friends, biophysicist Paul Greengard and his wife, the sculptor Ursula von Reidingsford, helped by other supporters of this great institution. Paul has spent much of his Nobel Prize-winning career studying how nerve cells communicate with each other in our brains. Ursula is a distinguished artist whose works include monumental cedar-based sculpture with subtle textured surfaces. Paul and Ursula both combine creativity with compassion in their work and in their lives. And as this prize makes clear, they also know that women need to work, to create, to find meaning in life, and work just as much as men do. This prize signifies their shared sense of the importance of discovery and the need to give women their chance at it. Thank you, Paul and Ursula. I've spent my life as an art collector and as a philanthropist. That has happened to me with the help of an art-conscious family and an art-rich community. My father collected Western art. My mother was a concert pianist. All four of my siblings have strong interest in the arts. My brothers in architecture, sculpture, and film, my sister in theater. Growing up, I had a deep and constant relationship with the Cleveland Museum of Art. I have a relationship still, even today, with that great museum of my girlhood. I think I learned during my childhood years how art can teach and how museums can provide energy and solace to young artists. These lessons have lived with me since. Certainly they were with me years ago when we started Studio in the School, a program that puts artist instructors in New York City public schools. and when my foundation created internships in museums for minority college students, and when we helped foster an alternative spaces and museums in minority communities. I remain mindful of these lessons today when we find cultural projects in the Art for Justice initiative. In school and college and in graduate school, my most important mentors, many of them women, nourished my love of art and my love of helping others. I have been so fortunate to be able to collect and enjoy the work of painters and sculptors, printmakers, and photographers. I have been fortunate to be able to support museums and other institutions to share works of art with the public through loans, exhibitions, and publications and education. In collecting art, I have long had a particular interest, a special interest in the work of minority artists, including women, and yes, women artists are a minority. Compared to men, few women get attention from the art world. Those who are recognized are seldom given solo shows or featured in group shows. The work of women artists sells for less and much less. Women are seldom ranked in auction choices. There are few of them in major collections. Dealers will say that a woman's subjects, even their materials, are feminine rather than universal like those of men. Most upsetting, really, is how deep the kind of bad reasoning is. As the artist Elizabeth Murray famously said, Cézanne painted cups and saucers and apples, and no one assumed he spent a lot of time in the kitchen. I do believe that today those of us who support women artists, including smart professionals in museums and galleries, and some art schools and residencies, are making inroads against this kind of bias. But there is a long way to go before women are secure, well-received, and fairly treated in the world of visual arts. We have a lot to do. I see a startling convergence in science and the arts. That is the need in both fields to ensure that women of talent are acknowledged and included and advanced. When I find myself asking what the roles and responsibility of talented, education, ambitious women in other fields, are women underrepresented only in art and science? Is talent obscured by gender? Is the workplace fair for women leaders? What can our girls expect as they grow up in today's workplace? There are few people I find who are concerned about this and reporting on it. In Forbes magazine, Janet Neapolitan, president of the University of California, provides sobering facts about women in higher education. Today, women make up more than 6% of college students countrywide. But once in the job market, these college education women earn 80 cents of every dollar a man earns. Neapolitan writes, the gender pay gap has narrowed by less than half a cent a year since 1963, when the Equal Pay Act was signed for minority women. The ratios are worse, ranging from 54 cents to 63 cents for every male dollar. To put it simply, a woman with a bachelor's degree earns the same as a man with an associate's degree. When the giant business software company Salesforce invested millions to achieve salary parity across their workforce, they found to their amazement that equity actually returned. A significant factor was advancement practices, which favor men. A recent study by the American Association of University Women finds a sad finding to this sad story. College women graduate not only earn less on their jobs, women college graduates graduate with more student debt than men do. Women hold nearly 2/3 of the outstanding student debt in the country. In short, women college graduates earn less and owe more. On a different note, though, also a serious one, Forbes recently offered 8 unbelievable gender pay gap statistics. They looked at top athletes, top actors, and top CEOs. The 100 highest earning athletes are all men. Among actors, only one woman appears in the list of the 10 highest earning in the field. More telling, perhaps, is that highest earning actresses earn 24.8 cents on the dollar to pay to actors. The Forbes list of the 25 top leading hedge fund managers and traders are all men. And the 25 richest people in tech are all men too. In modeling, though, the top earners are all women. About another field, the New York Times reported last week in an article about the rise of women in architecture that fewer than a third of the members of the American Institute of Architects are women, and that the first time a woman won the Institute's gold medal was 2014. Overall, the Times reports in this article about improvement in women in the field. Women occupy just 10% of field highest ranking jobs. I once sat on an architecture search committee, and when it came to a choice between a man and a woman, they chose the man over the woman, and her background and her size was actually called into question. There was only one man who sided with me in recognizing her accomplishments. Politics, especially as we look ahead to midterm elections, there's considerable talk about the surge of women running for office. The 2018 midterms have set a record for the number of women candidates who have filed and for the number who have won primaries in House and Senate seats. The number of women winning primaries for the governor's office is also the highest ever, but reports the Times, it is possible that the number of women actually in office will remain the same or even decline. This is possible and even likely because women are running in House districts that favor the other party. And many women are challenging incumbents who almost always win. Exceptions for women running for governor are modest. One acknowledged observer is quoted, "When a woman is running to be the CEO, voters need more evidence, traditionally, to see that she's prepared for the role than they do with a man." In fact, it seems that we want more evidence that women can do the work, whether we are talking about politicians or tennis players or architects or scientists or artists. What that means, of course, is that it is very much more difficult for talented women, whatever their talent, to find their true and merited places in the complex and comparative environment at the top of our heap. We must do what we can to face this and find ways to change it. One way to face this and change it is to acknowledge and salute accomplishments that outstanding women make. And that we do here tonight. Professor Jennifer Doudna, our honoree tonight, first made her name working on the structure and function of RAN, a cousin of DNA, which helps, I'm sorry, but I don't understand this too well, but I will try, which helps catalyze the chemical reactions of proteins. She has had a long career working out the chemistry underlying RNA's many biological function, motivated by the possibility that RNA might provide some clues about the very origins of life, and hopefully that these discoveries can help us address specific infections and diseases. What intrigues me and inspires me is that Dr. Doudna has discovered so much of her time to considering the ethical implications of how this science will be used. What also intrigues me is that the professor has thought of like a scientist since she was 13 years old. When she discovered biochemistry, she said that her father gave her the James Watson book about discovering the double helix of DNA. Her parents both inspired her, as did James Watson. She's also said that an important influence on her as a youngster was a lecture she heard at the time by a young woman scientist about cells and cancer. As an undergraduate at Pomona College, she was encouraged by Professor Sharon Bansonek, Women Research in Science Research, a woman successful in science research. These early encounters with women, the encouragement of her parents, her own determination, led our honoree to high hopes and high places. It is a signal pleasure to welcome her tonight as the recipient of this year's Meister Greengard Award. Congratulations, Professor Doudna, and thank you very much for all you do for the sciences. Thank you. Now, I present the award with you.
Yes, you want to take a seat? I have to introduce Jennifer now.
Okay.
Thank you so much, Agnes, for those really moving words. It was just terrific. That was just great, and I now would like to introduce Jennifer Doudna, our honoree this evening. Jennifer is professor of both molecular and cell biology and chemistry at the University of California, Berkeley, where she also holds the Li Ka-shing Chancellor's Chair in Biomedical and Health Sciences. She's an investigator of the Howard Hughes Medical Institute, a Gladstone Institute senior investigator, and the executive director of the UC Innovative Genomics Institute. Jennifer was born in Washington, D.C., and moved to Hilo, Hawaii at age 7, where she went on to graduate from Hilo High School. I fondly recall a meeting Jennifer and I had as part of a delegation from the National Academy of Sciences, meeting with a bipartisan group of U.S. senators to discuss the links between public support for basic science and advances in human health. None other than Mazie Hirono, who until last week might have been a relatively unknown senator. She was then a first-year senator from Hawaii, was in this small group, and she was absolutely enchanted to hear that a Hilo High graduate was such an accomplished scientist. The seed that was planted on that day came to realization and fruition just a few weeks ago, if I could have the slide up, as Jennifer returned to the University of Hawaii Hilo to deliver a distinguished lecture. This is just one of many accolades that Jennifer has had, and underscores the point that she's not only a scientist, she's been very involved and engaged in the public discussions about science and about its applications for human health. But as noted by Agnes, Jennifer was not always interested in science. In elementary and middle school, she found it a dry subject. Textbooks gave her the impression that it was simply a bunch of facts to be memorized for a test. And this changed when she was given a copy of The Double Helix by her father about the race to discover the structure of DNA. She then realized that research was a human endeavor with the potential to discover deep truths about nature, and she became intrigued. Nonetheless, she was only aware of one woman scientist, and that was Marie Curie, who had been born 100 years earlier. In high school, though, she had an inspiring chemistry teacher. She also went on mushroom hunts with a true aficionado and family friend, Don Hems, and they discovered previously unknown species of mushrooms. And that experience of learning something new that nobody had known before really caught on with Jennifer. She went on to earn her undergraduate degree in biochemistry at Pomona and then went to graduate school at Harvard in 1985. And there she heard about a young faculty member, Jack Szostak, who was devising experiments to explore the possibility that the origins of life mapped to the nucleic acid RNA. RNA had long been known for its role in biology's central dogma, in which segments of DNA and chromosomes are copied into short-lived RNA copies that direct the synthesis of specific proteins by ribosomes. A key attribute of these proteins is their role as enzymes, catalyzing the chemical reactions in cells that are required for life. In 1982, Tom Cech made the highly unexpected discovery that proteins were in fact not the only biological products capable of catalysis. Certain RNAs could perform catalytic functions as well. This surprising finding of RNA-based enzymes, or ribozymes, had profound implications. One of these concerns the origins of life. How did life get started? One critical element would be a self-replicating nucleic acid, and RNA seemed to be a good candidate. As a student in Shostak's lab, Jennifer showed that a ribozyme involved in RNA splicing could be re-engineered to splice together short RNAs using a complementary RNA template, thereby producing copies of the template RNA. This was a technical tour de force and a hint of bigger things yet to come. During her work as a graduate student, Jennifer had begun wondering, how exactly do ribozymes catalyze these reactions? The key, she knew, must lie in how they fold up, and their three-dimensional structure would establish the precise positioning of chemical groups that would hold substrates in the right orientation to allow their catalytic activity. She went to Tom Cech's lab for postdoctoral research to pursue this problem. By that time, many scientists had tried unsuccessfully to generate high-resolution structures of large RNAs. Conventional wisdom held that it simply couldn't be done. Large RNAs wouldn't form regular shapes that would crystallize, thwarting efforts to determine their structure by X-ray diffraction. But Jennifer was undaunted and determined to crack this problem. And after working through a lot of experimental challenges and failures for the first 2 years, getting only crystals that gave fuzzy diffraction patterns, she eventually put new crystals on the X-ray beam, and the X-ray diffraction pattern suddenly revealed a beautiful pattern indicating a sharp diffraction that was going to yield resolution at very high resolution, giving her confidence that the structure could finally be solved. At that time in 1994, Jennifer joined the faculty at Yale and set up her own laboratory. There she completed the structural analysis, which she published in 1996. This included novel structural principles of how RNAs could fold and stack, and the structural principles that she uncovered, although novel at that time, have been found to play a role in a wide range of important RNA molecules. At Yale and at the University of California, Berkeley, where she moved in 2002, Jennifer has continued her quest to understand how RNAs contribute to a wealth of biological activities. Her biochemical and structural biology studies have shown how the hepatitis C virus co-opts the protein manufacturing machinery of mammalian cells, how an enzyme called dicer creates small RNAs that can destroy messenger RNAs, those RNAs that contain instructions to make each protein in the cell. She also determined the structure and function of the signal recognition protein, which identifies newly synthesized proteins exiting the ribosome that are destined for export to the cell surface or the endoplasmic reticulum of the cell. These were extraordinary contributions that any scientist alone would be enormously proud of. The combination of them made Jennifer, by this time, easily a wonderful candidate for the Meister Greengard Award. Nonetheless, Jennifer's profound understanding of RNA structure and protein interaction over these 20 years of her research left her uniquely poised to build upon the discovery of a bacterial immune system called CRISPR-Cas, which protects bacteria from invading viruses and plasmids. Luciano Marafini, here at Rockefeller University and here with us tonight, showed that the CRISPR system, surprisingly to many, actually attacks DNA of these invading viruses, and a finding that was unanticipated. Jennifer and colleagues in 2012 then went on to assemble all the parts necessary to make this work in vitro, and doing beautiful biochemistry in 2012 showed that the CRISPR-Cas system acts by making double-strand DNA breaks targeted to specific sites by guide RNAs. These provide signals to the cell for DNA repair, which allows the possibility of repairing with just about anything you want to put in its place, providing the idea that this could then be used for gene editing. This was a seminal experiment in the development of CRISPR as a gene editing tool. The technology since then has simply exploded, making gene editing an all-purpose tool for biomedical research, to knock out genes in cells or organisms, to introduce specific mutations into particular genes, to tune the expression of particular genes to higher or lower levels, for imaging the location of specific genes or transcripts in cells, and many other applications that are still being developed. CRISPR technology also has tremendous potential for application in plant science, for example, readily producing drought-resistant crops, and also for human health. And indeed, initial clinical trials are already beginning to attempt to bypass or correct disease-causing mutations in people. These tools have rapidly disseminated into wide experimental use and are truly transforming biomedical science. Jennifer is a member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Science, and a foreign member of the Royal Society. Jennifer has received an incredible string of honors in addition to the Meister Greengard Prize, including the Breakthrough Prize in Life Sciences, the Gruber Prize in Genetics, the Canada Gairdner International Award, the Japan Prize, and many others. In 2015, Jennifer was named one of Time Magazine's 100 most influential people in the world. I hope you picked up a profile of Jennifer as you came in this evening to learn more about her incredible career. The profile was written by the wonderful science writer Evie Strauss, who is also with us this evening. And now, please join me in welcoming Jennifer Doudna to the stage. Right here. Can you come up here? So Jennifer, why don't you go to Agnes's right? Yeah. Do you have your text? Yes. Okay. So, it's now my pleasure to read excerpts from the citation that is being presented this evening. Jennifer Doudna, the Rockefeller University is proud to honor you as the recipient of the 2018 Meister Greengard Prize. You are being honored for your incredible discoveries about RNA structure and function, which have provided insight into the mechanisms of catalytic RNAs, revealed general principles that guide the folding of numerous physiologically important RNA molecules, unveiled critical aspects of RNA-protein interactions, and have contributed mightily to the development of gene editing using CRISPR. By solving the structure of a key portion of a ribozyme, a challenge many of your colleagues believed was insurmountable, you discerned the properties that allow this catalytic RNA to assume its three-dimensional shape critical for its enzymatic activity. You peered deep within this and other fascinating RNA molecules to reveal how they perform varied roles inside cells, including some that were long thought to be the purview of proteins alone, and others that allow RNAs to program double-strand DNA breaks at specific sites. You have set the highest standard for the community of scientists by demonstrating how curiosity, courage, and dedication can fuel the boldest discoveries. Today, in presenting you with the Meister Greengard Prize, we recognize and thank you for tackling daunting research problems, finding solutions that elucidate fundamental life processes, and hold great promise for improving human health.
It is my honor this evening to present Dr. Jennifer Doudna of the Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. On behalf of the Distinguished Selection Committee of the Meister Greengard Prize and all of us here today, we congratulate you on this award. Thank you.
Thank you very much.
you. Take that. Hold that up. Hold it. Agnes, come stand with us for a picture. Great.
Thank you.
The podium is yours.
Wonderful. Wow, this is an incredible evening. I am really honored to be here. It's a really special pleasure to be at the Rockefeller University, one of my favorite places, and a place that has inspired me in many ways over the years in science. I think we're about to have a conversation, Rick Lifton and I, with input from all of you, but I want to just very briefly, I hope you'll allow me to just say a few words of thanks, because there's so many people I would like to acknowledge. First of all, Paul Greengard, of course, and his wife Ursula, and the selection committee, and Rick Lifton here at Rockefeller. It's just a really great pleasure, and I thank you so much for recognizing me in this way. And I particularly am honored to be part of this litany of wonderful women who have been honored previously by this prize. So thank you deeply for that. I also want to acknowledge a lot of colleagues and friends. Many of them are here tonight. In particular, Corey Bargman, Charlie Rice, Seth Darst, Richard Axel, Luciano Marafini, Tizia DeLonga, Wayne Hendrickson. You've all been people who, I don't know if you know how much you have all influenced me in my thinking and inspired me over the years in many different ways. And there are many others that I didn't mention, but I really am deeply honored by the presence, your presence here tonight. I want to call out Sam Sternberg in the audience. Sam, you're representing my past and present lab members. Sam Sternberg, a graduate student at Berkeley with me, and now a professor at a little place down the road, Columbia. And it's just been a true honor to have the pleasure of working with so many wonderful students over the years, and I really feel this award recognizes their work and the work that we do together. I feel that science is a collaborative effort, it's teamwork, and that's really where I get a lot of my pleasure, honestly, is having ideas and working on them in the laboratory with wonderful people that have come to me over the years. And then finally, I want to thank my sister, Sarah Doudna, sitting here, and our cousin, Pat Doudna, sitting in the front row, representing my family, and of course, my spouse and my son, spouse Jamie Cade, and my son Andy Cade, who are not here tonight. But it's really my family that has given me the push I've needed at various times in my life to carry on with what I'm doing, have faith in myself, and pick myself up when I'm down. And it's a really great pleasure to be here in your presence this evening. So with that, thank you again.
Well, thank you, Jennifer. You know, I'll start by commenting that not having this wonderful audience here and not having you give a science talk to this audience strikes me as a bit like inviting Pavarotti and not asking him to sing. So I thought maybe we'd start by asking what's going on in the lab that excites you the most right now?
Well, I guess I would say two things. One is we're continuing to understand and investigate the fundamental biology of these CRISPR adaptive immune systems. And I think here at Rockefeller, you know a lot about this from Luciano Marafini's work. But I think there's a lot of very interesting basic biology still to be uncovered. And so we're having a lot of fun exploring the proteins and RNA molecules that are part of those pathways. The other thing we're doing increasingly is really more in sort of your realm of science, which is trying to figure out how we can use this gene editing technology to affect human health in positive ways. So we're very excited about opportunities to deliver gene editing molecules into cells and tissues, figure out how to do that efficiently, and also to understand the genetics of disease in ways that haven't been possible in the past.
Yeah, that's great. So one of the issues that you've been actively involved in is not just doing the science, but playing an active role in thinking about the policy that arises from it. I'm interested in your thoughts about the importance of scientists being actively engaged in the ethical discussions, and also how you think those ethical discussions are playing out.
I think over my time in science, we've seen a real destruction of trust in scientists and science in general. I think that's very unfortunate. I have to say, I think it's maybe partly scientists' fault that we need to get out of our labs a little more and tell people why we do, what it is that we research and why it matters. And we had this conversation today over lunch with some of the students and postdocs here. And I think that for me, this whole process of thinking about a developing technology, realizing that it was going to have very profound ethical and societal implications, and then trying to start educating myself about how to talk about it publicly has been a real learning experience. And I'm still on that trajectory, but I'm very optimistic about opportunities to move forward responsibly.
So when you and I were training, well, I trained a lot earlier than you did, but when we were in training and thinking about basic science, the opportunity to think about translation from basic science was in a much earlier phase of development. Your work in part started with tetrahymena, and I'm interested in your thoughts about as we spend $30 billion a year plus of public money for through the NIH, how we ought to balance the ratio of high-risk work in simple systems versus translational opportunities.
We clearly need to have both. I think what's happened over the years is there's been a real shift towards what we call translational research, meaning focusing on particular diseases and trying to solve and cure them. Which is clearly an important goal. I think the question is, how do you get there? And what I've found in science, and I think CRISPR is a great example of this, is that in many cases it's curiosity-driven fundamental science that uncovers things that none of us could predict. I mean, who knew that bacteria had evolved a programmable system for finding and destroying viruses? I mean, that was something that I think a few years back nobody would have anticipated. So. I'm just an advocate of supporting both kinds of science. The challenge is how to get that balance right.
Yeah, great. So as you think back on your early career, I'm interested in what was your interaction with your mentors like? To what extent were your mentors telling you, okay, now you do this, 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 and this, versus the free-range graduate student? Go into the lab, find something cool to work on, and learn about it. And I'm interested in what the approach was when you were a student and how that's translated into how you mentor students today.
Well, my first real research experience in the lab was actually when I was in college, and I was working with a wonderful scientist, Sharon Panasenko, and she had she trusted me to come into her lab one summer and start doing experiments. And I'd struggled a bit in general chemistry initially in college, and I wasn't very confident in my abilities in the lab. And what I discovered was that she gave me a lot of freedom in the lab, and she sort of said, here's a question, here's something we want to do, go figure out how to do it. And I realized how much fun it was, and I also realized that I could trust myself. Things didn't believe me, things didn't always work, but I could sort of make my way through experiments, and that started to give me the confidence to move forward in science. And then when I got to graduate school and I worked with Jack Szostak, as you mentioned, he was a very hands-off scientist in a sort of a daily, day-to-day kind of basis, but he also was still doing experiments himself in those days. He would come into the lab and he would be setting up reactions, and so he was always talking to me in the lab about science and how you do the right controls and things like that. So I also got the sense from him that he wanted my intellectual input, which was very important for building my own self-confidence. And then when I moved on to my postdoc, I was working with Tom Cech, another absolutely wonderful scientist. And there, that was sort of post-Nobel Prize for him. He was on the road a lot, and so we in the lab were really working very independently. And I had the joy of interacting with many smart people there and working together on problems and figuring out how to work through whatever experiments we were doing. And then in my own lab, I think partly influenced by my experiences with my mentors. I, have been, I think, and Sam Sternberg could maybe comment on this later, but, I think I've been a mostly very hands-off kind of mentor. I tend to I like to bring people to the lab who I feel are going to be great team members. I love team building and finding people that are going to work well together in a group setting in the lab. And then, brainstorming with them about what science we're going to do together, what problems we're going after, and then basically giving them the freedom to go after it and study it in the ways that they think are going to be best. And then, of course, working through the inevitable challenges that come up. So that's really what it's like in the research lab for me. It's it's very much a team effort, but I give a lot of trust to the people in the lab to use their intellect to address the kinds of problems we're going after.
So if you were advising young women scientists today, or aspiring women scientists today, thinking about mentorship, what advice would you give them in picking a lab? And maybe on the other side, since you trained with a number of very distinguished male scientists, what advice would you give men in science who are mentoring women in the laboratory? Any advice you'd offer?
Well, I said to the students today at lunch, that I think I've had the good fortune to work with a number of wonderful mentors over the years, men and women. And so I think that, what's been important for me, certainly, and I think for others, is to look for people who inspire you, who are the quality that, sort of do the kind of work and science that you aspire to do. I've always tried to work with people, whether they're coming to my own lab or people that I've worked for, that I think are better than me, smarter than me, people that I really admire deeply. And I think that's been a very valuable thing in my career, is just really to look for people that I can learn from by the kinds of examples that they set.
So as you think about the future of CRISPR technology, what do you imagine over the next 5 to 10 years coming from the field?
I think incredible things are coming. I really do. We're already at a point where clinical trials are starting with the CRISPR technology to cure various genetic diseases, and I think we'll see more of that. I think there's going to be a lot of fundamental discovery of the genetics of disease that will happen increasingly and will give new insights into both basic biology, but also opportunities to intervene therapeutically in ways that haven't been possible in the past, even eventually for complex diseases. And then I think, it's not mentioned as much, but, I think there's a lot of exciting work coming in agriculture and in synthetic biology in ways that, are hard to really predict, except that we know that now being able to manipulate plant and other kinds of organismal genomes is giving scientists a tremendous tool for understanding fundamentals of these organisms, but also manipulating the way that they interact in the environment.
Great. So if you were a graduate student today and just starting out, are there areas of science that you would be drawn to that you'd say, if I were starting today, this is what I'd do, other than the CRISPR field?
Well, I think, there's a tremendous opportunity right now in genetics and genomics. I'd probably come work for you, Rick, actually.
There have to be better things to do than that. But, well, I think we're drawing to the end of our time, regrettably, but this has been a really inspirational night for me, and I hope for all of you as well. Jennifer is truly an extraordinary scientist, and as I think you've learned tonight, an extraordinary citizen of the scientific community and of society at large. And we're just so incredibly proud to have you with us here tonight, and congratulations and thanks again.
Thanks.